All right. Hi, everybody. Welcome to the rest of the lab for this week. So, again, my apologies for not being able to cover this stuff during lab. It just wasn't possible with all the different things that we had to do. So, this is essentially a work through of what you will be doing with your gel pictures and how you will calculate the sizes of the unknown bands, right? And so the whole assignment for this lab is really kind of based around the process that you'll be following. And a big chunk of it is drawing your two graphs, right? 30%. Oh no, 30 points out of 50, so 60%. So that's why I want to make sure that you understand what you're doing because, I want you to get it right. So essentially what we have, or what you just did, is you ran a gel, right? And so what that means is essentially you have, gel, holes in it called wells, and you put your DNA in there, whether it's a marker or I can't really have a go at the person who put D on those tubes if I can't do it myself. Anyway, you put your DNA in those wells, and then you applied a voltage difference. So the big deal here is that DNA is negative, right? You put it at the negative end and you apply, oops, that's not going to work. You apply a voltage difference across, so that DNA gets pulled to the positive end. Now, you want to separate these bands, right? These different chunks of DNA based on their size. So you'd think, oh, hang on, sure, DNA is negatively charged and we, attract it to the positive end of the gel. Surely this will all just move through in a lump, right? So you're not going to be any better off than you were to begin with. However, this gel is actually pretty cool, like super crude, like agricultural type technology, but very, very effective. Because essentially what this is made with the same stuff that your McDonald's milkshakes are, right? This is an agarose gel. And essentially when you boil agarose and you melt it, when it sets, it forms a mesh. Kind of looks like that if we were to look at it in cross section. Or even this section, right? So when you have your different bits of DNA, let's use colors because I can. Like so. When you pull them to the positive side, essentially they vary in how quickly they make it through this mesh based on their size. So, and also based on the mesh pore size as well, which you can control within a range. But essentially the smallest one kind of whips its way through real fast, right? Effectively unimpeded by the gel itself. So the smallest is going to be down here. And then the next largest is going to be a bit further up. And then the next largest further up still. And the largest of all will take a very long time because essentially this one has to kind of weave its way in and round all the way through. And it just takes longer, right? So essentially what you have done is that you've separated different size fragments of DNA by size, essentially by how quickly they're able to migrate through this gel. So far, so good, right? However, the rate at which they do so is not linear. And so it's very hard to figure out exactly how big a fragment is. Now we can approximate, we're using molecular weight markers, and most of the time that's all you really need to do. Come on. And so just making sure I don't, let's get rid of that for a second. I don't know, we could put sizes to these. Now let's do that in a little bit. So you can add what's called a molecular weight marker, often called a DNA ladder because it looks like a ladder, stuff like that. And so this is a mixture of fragments of which the sizes are known. And they kind of look like so, give or take. Yeah, they vary from manufacturer to manufacturer. And let's say this is 10 kilobases. This might be 1 KB. This might be 100 base pairs, for example. So you can approximate, and usually that's all you need to do. Say you're amplifying something, it's like, yeah, that looks about right. Nothing similar to it, so you're good. But the problem lies is if this unknown band is between these. So this one, let's say this is 2, well, let's say that's 350, and whatever, it doesn't matter, right? So this one is going to be between 100 to 350 base pairs. And, you know, probably closer to 350. Let's say maybe 320. But you don't know, right? Because essentially what you're doing is you're interpolating between two known points and going, that's what that is, right? Which isn't a very scientific way to do things. Now, what you can do, which is the purpose of this whole lab, is essentially to create a value or relationship, I guess, between size and distance moved using something we already know. So essentially what you can do is you can work out, okay, 10 KB moves, let's say 1 centimeter. 1 KB moves, what would that be? 4 centimeters. And so on and so forth. This might be, I can't actually remember how big the gels were. Doesn't really matter, right? You get the idea. Right? And so now we can plot those fragments by size and distance traveled. Just try to remember which way around. I think it's size that's the log function. I should have read the lab handout again when I was busy eating my lunch. So we have, yeah, I'm pretty sure this is what it is based on memory. I don't know what that'd be. Base pairs, maybe. And this would be in centimeters. Right, so this is very important because this is not a linear relationship. You can actually see, right? You know, we've got essentially a log function here. We got 0.1, 1, and 10. And they're equally spaced. So that's telling you that the distance moved isn't a linear relationship with size, right? It's actually a logarithmic relationship. You don't have to worry about that because all you do is you use semi-log paper. So the size has to be in as a log function. And really what that looks like is you would have 10, 1, 0.1. Right? It just looks a little funkier, that's all. There's a copy of this in Blackboard. So either use that, draw it on the paper, like print it out, and take a picture of that to upload, or if you're going to do this in Excel, which is fine, do some funky stuff in Excel. This axis has to be log, logarithmic axis, which you can change in format axis, right? And then there's a box somewhere in there, you tick it because it says log. So essentially, this is going to be an inverse relationship. So 7, 8, 9. Yeah, that'll do. 1, 2, 3, 4. Man, this blue doesn't want to come off. 5, 6, 7, 8, 9. There you go. That'll do. Not going to do all of these because it's, same principle, but enough to give you the idea. 3 points, plot a graph. So we've got 1 centimeter, we have 4, and we have 8. Oh, look at that. Couldn't have done that better if I tried, right? So you're going to be doing that for all of these bands, or at least as many as you can accurately measure. As long as you measure from the same point for every single band. That's important, otherwise it's just not going to work. Once you've done that for the markers, you have 2 markers, so you're going to be drawing 2 graphs, and you're going to be plotting the figures for each marker. Again, based on size and distance traveled. Okay. Then what you do is you draw what's called a line of best fit, right? And so essentially what you want to do is make sure that the distance from your line to each point is as small as it can be for all the points, right? So you can't draw one like that, for example, which is great for these two, sucks for that one, right? Has to be as good as it can be for each point that you have plotted. And again, you need to do this for each marker, both the SL1 or the Edvitech marker and the Promega Kilobase marker, right? Then what you do, different color, is you measure your unknowns. So you have an unknown 1, 2, and 3, right? Different number of fragments. You have to do this for all the fragments. But at least now you only have to do it for like one set of measurements because you use both of those in that same set of measurement in both of the graphs that you're going to be drawing, right? So let's say this is the Edvitech graph, right? So I measure that, and that is 1.5 centimeters. That is, I don't know, 3.5, and so on. That would be 6, maybe, whatever, right? Get the idea. So now we have a relationship here between size and distance so that we can figure out size for any size fragment, ideally between these two extremes, as long as we know the distance that they have traveled. So for our largest unknown, which has moved 1.5 centimeters, right? That's exactly there. It's kind of like the t-test, not t-test, chi-square graph. You go up until you hit the line, and then sideways until you hit the size axis. And that would be a better labeled axis than what I have, but let's say that is 8 kb, right? 8,000 base pairs. You do the same for the next one, 3.5, go up. That would be, say, 1.5 kb, and so on. For every single unknown, that will be, I don't know, base pair. Kind of starting to fall apart a little bit right now, but that will do. And that's how you figure out the size of the unknowns. Now, because our two markers differ in the sizes of the fragments and how many fragments there are, one marker may be better than the other for drawing this graph and calculating that relationship. What I want you to do is to figure out the sizes of the unknowns using the two different graphs and basically make a judgment which marker is better and why. That's the last question on the assignment. And that's it. Now, obviously, I might have not answered all the questions that you might have, or some of this might not make complete sense. So please do email me or next lab after the lab, probably because it's going to be just as bonkers as this one. Come and ask and I'll help you work through any of the questions that you have. I'll take a picture of this and upload it to Blackboard as well as posting this to YouTube. All right. Have fun, everybody. I'll catch you later.